My name is Lila Wells, and my project is carboxyl-functionalized multi-walled carbon nanotubes and PTPN22 antibodies in detecting abnormal antigen levels for immunosensor applications. 1 in 5 Americans suffers from an autoimmune disease. My little sister is that 1 in 5, and she suffered for months before being diagnosed with type 1 diabetes, which is caused in part by the dysregulation of the PTPN22 gene. Blood tests, the most common autoimmune diagnostic tool, are daunting and not easily accessible to many people. This led me to brainstorm alternatives. Carbon nanotubes were used to detect cancerous tumors in the past, but their high conductivity and ability to be functionalized has yet to be applied to autoimmune diagnostics. As PTPN22 dysregulation leads to abnormal antigen levels in the body, this project aims to detect antigen levels through changes in conductivity of a nanotube antibody complex. I took a course in research and experimental design my junior and senior year that involved an independent study. My professor, Dr. Freeman, mentored me along the research process, although my experiments were self-directed. I began my experiments in May of 2018 and completed all research in my school's lab, except for FTIR testing at a Yale lab. I reviewed McDevitt's study and observed the use of a nanotube antibody complex to sense cancerous tumors. Also, Arkin's study used complex gold nanoparticles with a nanotube electrode to detect HER2 protein levels in the body. My project builds on previous research by exploiting the conductivity of carbon nanotubes in a nanotube antibody complex in order to synthesize a cheap and simple biosensor. In the lab, I first ground nanotubes and mixed them into 5 different solutions, varying in nanotube length and diameter, as well as the amount of antibody present. I pipetted drops of each solution onto filter paper to synthesize the physical biosensor. I then tested the resistivity of each sensor strip before and after immersing it in a solution with simulated body fluid and differing levels of PTPN22 antigen to simulate healthy cell environments and those afflicted with autoimmune disorders. Resistivity, which is inversely tied to conductivity, was measured using a multimeter. I completed this procedure again with plasma carbon nanotubes that had a higher carboxyl functionalization and were more likely to bind to the antibodies. I used FTIR spectroscopy to observe if the nanotubes had bonded with the antibodies and if sufficient carboxyl groups were present in the nanotubes. Tentative results corroborate the idea that the sensor strips comprised of carbon nanotubes and PTPN22 antibodies effectively sense changing levels of PTPN22 antigen through shifts in resistivity and conductivity. Another set of trials with plasma nanotubes is set for mid-January to confirm these observations. Thank you so much for your time.